So picture this. A water molecule swoops in and literally cleaves the bond between two rings. Did you know that mastering section 2.2, glycosidic bond breakage by hydrolysis, actually accounts for roughly 15% of your total marks in paper 2 of the Cambridge Biology 9700 exam? That is huge. Welcome to the Explainer. Today, we are completely deconstructing the exact biological mechanisms and, more importantly, the analytical strategies that senior examiners are actively looking for. We're just going to skip the fluff today because this topic, it's an absolute heavyweight. Think about it. 15% can literally make or break your final grade. And the crazy part is, even when students know the raw biology, they've memorized the textbook, thousands of them still drop these crucial points. Why does this happen? Well, it's usually because they focus purely on memorizing those facts instead of figuring out how examiners actually test the application of those facts in the real exam. Just look at this direct quote straight from the principal examiner reports. It perfectly hits the nail on the head. It says, candidates consistently confuse cause and effect. And, get this, forget to check units and terminology. So they end up losing these grade-defining marks before they even flip the page. It's so frustrating to see, but today we're going to fix it for sure. All right, let's dive into Section 1, Mastering the Core Principles. Okay, so here's the deal. Examiners explicitly prioritize analytical depth over superficial breadth. What does that mean for hydrolysis? Well, it means you absolutely have to map out the precise sequence of events. Just writing down water breaks a bond is definitely not going to cut it. You have to explain exactly how the addition of a water molecule specifically cleaves that oxygen bridge, which then result in two totally separate monosaccharides, each with their own distinct hydroxyl groups. You've got to be specific. Now, the assessment objectives for this, they target 4 distinct areas. It's kind of like a giant puzzle. The principal examiner reports actually highlight that the candidates who score the highest marks are the ones who reference cross-topic connections. Meaning, they integrate their knowledge of molecular hydrolysis with other areas of the syllabus, like enzyme action, for example. You gotta connect the dots. So what is the hidden examiner clue that changes your answer entirely? Well, in just a minute, I'm going to share that specific clue with you. It's literally the detail that separates the top-tier candidates from everyone else. But hold your horses, because first, you really need the procedural framework that's going to make that specific clue super obvious when you're actually sitting there reading exam question. I'm moving right along to Section 2, Systematic Problem-Solving Framework. The absolute crucial point here is what we call the deconstruct, solve, and evaluate sequence. Seriously, whenever you face a really complex problem involving glycosidic bonds, you have to use this exact workflow to guarantee maximum marks. So, step 1, identify the key given information. Step 2, break the whole thing down into logical steps using your biology principles. Step 3, solve it systematically with super clear working. Step 4, you've got to check for reasonableness. Does your answer actually make sense? And finally, step 5, make justified recommendations based on what you found. And let's not forget, your data interpretation skills are absolutely critical here. When you're analyzing hydrolysis experiments, say you're measuring the changing concentration of reducing sugars as a polymer breaks down, you have to extract that data and calculate the right statistics. This framework lays it right out for you. You're going to use a t-test if you're comparing means, you'll use chi-squared for categorical data, and you'll go with correlation tests if you're looking at relationships between variables. It's a lifesaver. Furthermore, doing practical work for hydrolysis really develops your experimental skills. And trust me, examiners test these rigorously. You gotta follow these exact sequential instructions. First, select the appropriate apparatus. Then, make accurate observations and effectively record your data in suitable tables. But wait, don't just stop there. Top band students go the extra mile. They actively identify anomalies in their data and they draw solid evidence-based conclusions from what they've actually found. All right, Section 3, the flawed answer breakdown. Okay, pay super close attention to this examiner alert because it is a massive red flag. The biggest warning we get from examiners is that students' answers are frequently just not specific to the given context. Candidates are constantly confusing key terminology. So the golden rule is simple. Always define your terms before you use them in extended responses. Like, you need to clearly distinguish between an alpha and a beta glycosidic bond before you start rambling about them. Let's compare a flawed answer to an improved high-mark answer. A flawed answer is kind of built like a fragile house of cards. It just lists superficial similarities without data and totally confuses cause and effect. On the flip side, an improved top-tier answer, that's built like a solid brick wall. It uses comparative connectives to highlight both similarities and differences. It actively identifies the limitations of the evidence, and it supports every single claim with hard data from the source. Night and day difference, right? Which brings us to Section 4, High Mark Exam Strategy. By aggressively evaluating the limitations of the evidence you're given, your answers are going to instantly jump to the highest mark band. Just make sure you avoid confusing cause and effect, and I can't stress this enough, always check your units and your terminology. You also want to tailor everything to the very specific context of the question. Ask yourself, okay, what assumptions are being made here? Are there alternative interpretations of this data? And how do those alternatives actually verify or change my final conclusions? Here is a fantastic memory hack to help you retain this really complex procedural framework. Create your own personalized mnemonic right now. Seriously, grab a piece of paper and use the first letters of the key terms we just talked about, like D for definitions, C for causal relationships, and E for evidence. Actively building your own memory framework is a scientifically proven way to make sure you can actually retrieve this stuff when you're under the intense pressure of the real exam. So you now have the ultimate framework for evaluating glycosidic bond breakage by hydrolysis. You know exactly what areas the examiners are targeting, but leaving right now means you are completely exposed to the one hidden mistake that absolutely ruins otherwise perfect biology answers. There's a bit of a diagnostic cliffhanger here that we absolutely have to address. So what is that hidden cause of lost marks? It's the command words. Yep. Do you know the exact command words that decide your mark band? To uncover this hidden examiner clue, your mandatory next step is to watch our next explainer on those very command words. And hey, make sure to subscribe to Ace the Exams at acetheexams_IGCSE so you can absolutely crush your Cambridge exams. Until next time, I want you to ask yourself, if hydrolysis breaks the bonds that build our biological world, what processes are happening inside your cells right this very second to put them back together?